Hello, okay, let's take a look at Chapter 2, Cells and Cell Division. So the chemistry of cells. Cells are constructed from four classes of large molecules called macromolecules. Carbohydrates include small sugars and large polymers of sugars. Lipids consist of fats and oils, phospholipids and steroids. Proteins are polymers of amino acids and carry out a multitude of functions and activities. And nucleic acids are polymers of nucleotides. They store and transfer genetic information in the cell. Here you can see the main biomolecules in cells. The carbohydrates are broken further down into the subclasses of monosaccharides, which are your simple sugars, the oligosaccharides, short chain carbohydrates, and the polysaccharides, which are your complex carbohydrates. Examples of the monosaccharides are glucose and their primary functions as an energy source. The oligosaccharides, the main example would be sucrose, which is a common sugar, and the polysaccharides would be things like starch and glycogen, and they're used for energy storage. With lipids, you have the glycerides, which are glycerol plus a fatty acid. The phospholipids, which are glycerol, fatty acids, and a phosphate group. And the sterols, which are carbon ring structures. So the glycerides you primarily will see as fats, and they're used for energy storage. Phospholipids, an example there would be lecithin, and it's a structure of the cell membrane. And sterols would be your cholesterol. So you can see sterol there and cholesterol, sterol being part of the word cholesterol. And membrane structure and a precursor to steroids and hormones. Third class is proteins. You have mostly fibrous, which are sheets of polypeptide chains, mostly water, and they're insoluble. And mostly globular, which are protein chains folded into globular shapes, which are mostly water soluble. The example of the mostly fibrous would be keratin and collagen, which you would see in the structure of hair and structure of bones. And the mostly globular you would see as enzymes, hemoglobin, insulin, and antibodies. Enzymes function as catalysts. Hemoglobin provide oxygen transport throughout the body. Insulin acts as a hormone, and antibodies provide immune system defense mechanisms. The fourth class is the nucleic acids. You have adenosine phosphates, nucleic acids, which are polymers of nucleotides. Examples of these would be ATP, DNA, and RNA. And the function of ATP is as an energy carrier, and the function of DNA and RNA is for storage and transmission of genetic information. Cell structure reflects function. The cell is the basic unit of structure and function in all organisms, including humans. Although cells can differ in their shape and size, function, and life cycle, at the structural level they're still very similar. All cells have a plasma membrane, a cytoplasm, membranous organelles, and a membrane-bound nucleus. The function of a cell is under genetic control, and many genetic disorders cause changes in these cell functions. So here you've got the outline of a cell. You can see most of the major structures here. For instance, to the right in the purple, you've got the nucleus, which is made of the nuclear envelope, the nucleolus, chromatin, and the nuclear pores. Around that, you'll see some free-floating ribosomes and the endoplasmic reticulum. The smooth and rough endoplasmic reticulum in particular. If it's smooth endoplasmic reticulum, it just means that there's few to no ribosomes located on it. If it's the rough endoplasmic reticulum, it just means you have some endoplasmic reticulum membrane-bound ribosomal units. Then you have the Golgi complex there in the lower right corner. You have lysosomes and vacuoles. You'll see centrioles located throughout. You'll see those funny looking kidney bean-shaped structures called mitochondria. And of course, you have the outer plasma membrane and the cytoplasm inside the cell. Remember, a cell is a round or somewhat spherical 3D shaped structure, so it's not always like a flat surface like they show it in the textbooks. And I think the slide does a good job of showing that it's got depth to it. At any rate, that is the cell as a whole. Okay, so keep in mind, many genetic disorders alter these cellular structures. All these organelles, what they do and how they function, can be altered by these genetic disorders, causing different things to occur than what would normally be the case. So two cellular domains, the plasma membrane and the cytoplasm. There's a double-layered plasma membrane which separates the cell from the external environment. The membrane controls the movement of molecules into and out of the cell. You have molecule, which is a structure composed of two or more atoms held together by chemical bonds. When you see things like this in blue in the PowerPoint slide, generally means what's following is a definition. The plasma membrane encloses the cytoplasm, which is a complex mixture of molecules and organelles. And an organelle is a cytoplasmic membrane enclosed structure with a specialized function. So here you go, you can see the lipid bilayer here. You have various membrane-bound proteins that are embedded in the membrane. You have some carbohydrate chains that are sticking off of the proteins that are bound into the membrane. You can see the phospholipid molecules here with the hydrophobic head and the hydrophilic tails. And then you have cholesterol molecules in there embedded. You can see those in the yellow, the teardrop-shaped structures there off to the left side. Excuse me, I think I said hydrophilic head and hydrophobic tails. So the heads and the lipid, phospholipid molecules, they should be, let's see, hydrophobic heads and hydrophilic tails. The tails love water, and the hydrophobic heads do not. So sorry if I said that backwards initially. Caught myself though. Okay, organelles. Endoplasmic reticulum, the ER, system of cytoplasmic membranes arranged into sheets and channels. You synthesize and transport gene products here at the endoplasmic reticulum. The ribosomes are cytoplasmic particles that aid in the production of proteins. So here's a picture of the endoplasmic reticulum, the rough ER. On the right side there, you can see you have a photograph taken through a microscope. You can see the ribosomes embedded on the cross section of the ER lumen, and the ribosomes that are attached to the rough ER are really what give the rough ER its name. It's not just endoplasmic reticulum. It looks studded like or like sandpaper with a rough surface, and that's because all those little rough parts are ribosomes that are sticking off the surface of the ER. Organelles. You have the Golgi Complex, membranous organelles composed of a series of flattened sacs. The job of the Golgi Complex is to sort, modify, and package proteins synthesized by the endoplasmic reticulum. And the lysosomes are membrane-bound organelles that contain digestive enzymes. Keep in mind, Gaucher disease affects lysosomal function. It's one of the diseases that's highlighted in the book. You can read about that there. And let's see here. What you have going on here, this is kind of showing the mode of transportation of how things move from one location to the next, especially here at through the Golgi complex. So I'll show you over here. So you have the rough endoplasmic reticulum over here, and what you do is when these ribosomes make copies of the, once these ribosomes make the proteins that they're going to make, they generally go inside the ER lumen. So these ribosomes here, they're the same as the free-floating ribosomes that are out in the cytoplasm, except that these that are bound to the lumen tend to modify and make things that are going to enter into the lumen, where they'll get modified a little bit further and packaged up into these transport vesicles, and they will bud off of the endoplasmic reticulum, and then they will travel and fuse into the Golgi complex here. The membranes will actually fuse, and then they'll move inside, and then once they're inside, they'll get further modified by the Golgi complex, which that in turn will also bud off as those pieces are appropriately modified, and then they can move off as and be digested via lysosome, or they could go and fuse with the plasma membrane for to leave the cell, for instance. So lots of things can happen once it leaves the Golgi complex. But again, the purpose of the Golgi complex is to sort and modify and package, further package the materials that come from the endoplasmic reticulum. Okay, mitochondria are membrane-bound organelles in the cytoplasm of all eukaryotic cells. They are the sites of energy production. So here's a cross-section of a mitochondria, and mitochondria are also unique because they also have their own DNA, and we'll probably talk about this a little bit later if we get into the endosymbiont theory at all. But again, mitochondria also have their own DNA apart from the DNA in the nucleus of the cell itself. So there's a probably a transmission microscope picture of a mitochondrion. Okay, nucleus, membrane-bound organelle in eukaryotic cells that contains the chromosomes. And here are some pictures of the nucleus. To the bottom left there, you see all those things. It looks like the surface of the moon with all the craters. Those are actually nuclear pores. Then to the right, you have another type of cross section there. You can actually see, in the photograph to the right, you can actually see all those little things that look like worms are actually cross sections of the lumen of the endoplasmic reticulum. Okay, the nucleolus or nucleoli, if you're singular, it's a dense nuclear region that functions in synthesis of ribosomes. And chromatin is DNA and protein components of chromosomes. They generally appear as visible clumps or threads in the nuclei. Chromosomes, thread-like structures in the nucleus that carry genetic information. There are 46 chromosomes in human cells, which is the diploid number 2n. The haploid number would be 23, or half that value. And you'll find these in most human cells. The genes are fundamental units of heredity present on chromosomes. Sex chromosomes. Human X and Y chromosomes are involved in sex determination. And the autosomes are the chromosomes other than the sex chromosomes. So the human chromosomes 1 through 22 are the autosomes. And then that last set 23, is your sex chromosome, which will either be XX or XY, usually. Okay, again, there's that same slide with the overview of the organelles. We'll just go ahead, since we already looked at that, continue on. 2.3, the cell cycle. The cell cycle is a sequence of events that takes place between successive mitotic divisions. You may remember meiosis and mitosis from biology at some point previously in your life, or maybe not, but we're going to take a look at those briefly here in a moment. Okay, the three phases of the cell cycle. You have the interphase, which is the period of time in the cell cycle between mitotic divisions. You have mitosis itself, which is a form of cell division that produces two cells, each having the same complement of chromosomes as the parental cell. And then finally, the cells have to break apart, so we call it cyto or cell. Cyto means cell. Kinesis means to move, so it's literally translated as cell moving apart. And it's the cytoplasmic division phase of cell division, which is when the two cytoplasms, or the shared cytoplasm, actually splits into two separate cytoplasms to make the two new cells. Okay, overview of cell organelles. The nucleus is structurally it's a round or oval body surrounded by the nuclear envelope, and it functions to contain the genetic information necessary to control cell structure and function. DNA contains hereditary information. The nucleolus is a round or oval body in the nucleus. It contains DNA and RNA, and it produces ribosomes. The endoplasmic reticulum is a network of membranous tubules in the cytoplasm of the cell. Smooth ER contains no ribosomes. Rough ER is studded with ribosomes. The smooth ER is involved in producing phospholipids and has many different functions in different cells. The rough ER is the site of synthesis of lysosomal enzymes and proteins for extracellular use. The ribosomes are small particles found in the cytoplasm. They're made of RNA and protein, and they aid in the production of proteins on the rough ER and the ribosome complexes, known as polysomes. Okay, the Golgi Complex is a series of flattened sacs. They're associated with vesicles. They sort, chemically modify, and package proteins produced on the rough ER. The secretory vesicles are membrane-bound vesicles containing proteins produced by the rough ER and repackaged by the Golgi complex. They contain protein hormones or enzymes. They store protein hormones or enzymes in the cytoplasm, awaiting a signal for release. The lysosome is a membrane-bound structure containing digestive enzymes. These can combine with food vacuoles and digest materials engulfed by cells. Finally, you have mitochondria, round, oval, or elongated structures with a double membrane. The inner membrane is extensively folded. It's called the cristae. Its function is the complete breakdown of glucose, producing ATP. Okay, so here you got your overview of mitosis. You can see cells tend to be in interphase for quite a period of time, and interphase is broken down into the G1, S, and G2 phases. And then you enter into mitosis itself, which is that green section. And mitosis has 4 phases: P, M, A, T, PMAP: prophase, metaphase, anaphase, telophase. At which point the cell can then leave the cell cycle and enter into a inactive state known as G0, from which it can again come back to life at some point in the future, but it could go kind of dormant in a G0 state, or it could enter back into G1 and go through DNA replication and chromosomal replication, and finally preparation in G2 for division, and then re-enter mitosis again. So it can go around and around and around in this cycle, or not. So, interphase. Let's talk about those. I'll show you again here. Interphase. See the G1 there, that top left quadrant. Okay. G1 phase. Growth takes place after division. So, you had this previous cell division that's just happened, and now you're entering into G1, so growth is going to have to take place. The cell will double in size and will replenish organelles that were lost or depleted from the previous mitotic division, and also the same thing with the ribosomes. It will replace or repair and prepares for chromosome replication again. It'll then enter into S phase, which is the DNA synthesis stage. Copies of chromosomes are generated, and finally it'll go to G2 phase, which is the period of preparation for cell division. Okay, structures of the chromosome. You have a chromatid, which is one strand of a duplicated chromosome. It's joined by a centromere to its sister chromatid, and sister chromatids are 2 chromatids joined by the common centromere. Each carries identical genetic information. The centromere is the region of a chromosome to which the microtubule fibers attach during cell division, and the centromere location gives a chromosome its characteristic shape. It looks almost like a belt on the chromatids. So there you go. At the top you have one chromosome which is unreplicated, and then it goes down to one chromosome replicated. These are called sister chromatids at this point, and then you can see the centromere. It looks like that white button there in the in the middle, and that will attach to the spindle fibers. Okay, mitosis occurs in 4 stages. We said PMAP: prophase, metaphase, anaphase, telophase. So let's take a look at those. Prophase is the stage of mitosis when chromosomes become visible and contain sister chromatids joined at the centromere. And metaphase is the stage of mitosis when chromosomes move and become arranged at the middle of the cell. And anaphase, that's the stage of mitosis in which the centromere is split and daughter chromosomes begin to separate. And telophase is the last stage of mitosis. Chromosomes of the daughter cells decondense and the nucleus reforms. Okay, and finally you had to divide the 2 cells so they undergo cytokinesis, which in cytokinesis you get the formation of a cleavage furrow that divides the cell in 2, and then the organelles are distributed to the 2 daughter cells. Okay, so here we'll kind of go through this a little bit at first. Going up to A at the top, you have the cell at interphase. The cell duplicates its DNA and it prepares for nuclear division. Going down to B in the bottom left corner, you have early prophase. Mitosis begins. The DNA and its associated proteins have started to condense. The 2 chromosomes, which are color-coded blue, were inherited from the female parent. The other 2, the purple, are their counterparts inherited from the male parent. In step C, you have what's called late prophase. Chromosomes continue to condense. New spindle fibers become assembled. They move one of the 2 pairs of the centrioles to the opposite end of the cell, and the nuclear envelope starts to break down. And then in step D, transition to metaphase. Now the microtubules penetrate the nuclear region. Collectively, they form a bipolar spindle apparatus. Many of the spindle microtubules become attached to the 2 sister chromatids of each chromosome. Continuing on, step E. At metaphase, all chromosomes have become lined up at the equator of the fully formed microtubule spindle. At this stage of mitosis in the cell cycle, chromosomes are in their most tightly condensed form. F. Step anaphase. Attachments between the 2 sister chromatids of each chromosome break. The 2 are separate chromosomes and microtubules move towards opposite spindle poles. G. Telophase, there are two clusters of chromosomes which decondense. Patches of new membrane fuse to form a new form a new nuclear envelope. Mitosis is then completed. And then step I, interphase. Now there are two daughter cells, each is diploid. Its nucleus has two of each type of chromosome, just like the parent cell. Okay, so here's an overview of the phases of the cell cycle. You can go ahead and read these yourselves on the slide. I'm not going to go ahead and read them to you now, but this is definitely worthwhile knowing all these stages of all the phases and then all the characteristics of it. I would highly recommend you do that. Okay, 2.4, mitosis is essential for growth and cell replacement. Mitosis and cell division is an essential process. Human cells are genetically programmed to divide about 50 times. This is known as the Hayflick limit. This limit allows growth to adulthood and repairs, such as wound healing. Alterations in the limits to cell division can lead to genetic disorders, such as premature aging or to cancer. Keep in mind that cancer is Keep in mind that cancer is a disease of the cell cycle. Here you have a picture of progeria and premature aging. This is actually a 15-year-old boy, I believe. He was one of the longest living humans to survive progeria, but I think he did pass away finally in his 15th year. Causes premature aging, as you can see here. He's 15, but he looks like he's 80 or 90 years old, I guess. Okay, spotlight on cell division and spinal cord injuries. Many highly differentiated cells, such as those of the nervous system, do not divide. As a result, injuries to nervous tissue, such as spinal cord, do not heal through cell replacement. New approaches using embryonic tissue suggest that it may be possible to reconnect damage done to nerve tissue. And finally, newly discovered growth factors also show potential for stimulating the growth and division of cells of the nervous system. So cell division by meiosis, which is the basis of sex. Meiosis is a form of cell division that produces 4 haploid cells containing only one copy, paternal or maternal, of each chromosome. You go through 2 stages of meiosis, which are meiosis 1 and meiosis 2. In meiosis 1, members of a pair of homologous chromosomes physically associate. At this time, each chromosome pair consists of 2 sister chromatids joined by a common centromere. Each pair separates from each other into 2 cells, thus reducing the number of chromosomes by 1/2. So here you can kind of see the stages: prophase, metaphase, anaphase, and telophase 1. Because you go through meiosis 1 and meiosis 2, you'll actually go through PMAT twice in meiosis. PMAT 1 and PMAT 2. You'll see opportunities for things like crossing over and non-disjunction and such, and those will happen during meiosis. So feel free to read through the slide. I'm not going to go ahead and read through all this to you now, but you can certainly do that and spend quite a bit of time studying this slide, A through D. Now, in meiosis 2, you have metaphase 2, where the unpaired chromosomes align at the cell's middle. You have anaphase 2, where centromeres divide and daughter chromosomes move to opposite poles. And meiosis produces 4 haploid cells, N, each containing only one copy of each chromosome. In humans, the haploid number is 23, and the diploid number, or 2N, is 46. So here's meiosis 2. As you went through meiosis 1 a moment ago there with PMAT 1, we're now going through PMAT 2. And again, spend some time studying this, paying close attention to the pictures, and reading through each of the stages so you fully understand them. You'll probably see lots of questions related to these types of things on the test. Okay, chromosome number. The diploid number 2N. Each chromosome is represented twice as a member of a homologous pair. And haploid, each chromosome is represented once in an unpaired condition. Homologous chromosomes, there are maternal and paternal with identical gene loci. Chromosomes that physically pair during meiosis. And assortment, the gametes receive random combinations of maternal and paternal chromosomes. The result of this is meiosis 1. And meiosis produces new combinations of genes in two ways. One of them is random assortment of maternal and paternal chromosomes in metaphase 1. The chromosome pairs line up at random. And then part 2, crossing over, the exchange of chromosome segments between homologous chromosomes. So here they're showing in the very left picture, you have members of a chromosome pair, two sets of sister chromatids, one purple and one blue. And then each chromosome pairs with its homologue. So the purple one is the homologue of the blue one, for instance, and they pair up in the second diagram. And then the third diagram, the paired homologous separate in meiosis 1. And then in the very final picture, sister chromatids separate and become individual chromosomes in meiosis 2. Okay, here's the summary of meiosis. You can read through each of the steps and what kind of happens at each step. Again, another tool to be utilized. This is helpful. There's lots of good information on this for you. Okay, here's a comparison of mitosis and meiosis. And again, rather than getting you lost going through this, again, pay close attention here. You can see on mitosis at the very end stage, you have diploid daughter cells, whereas in meiosis, you have haploid daughter cells. And the reason in meiosis you want to have haploid daughter cells is because, remember, these are dealing with sex and sex chromosomes, so you want to have the appropriate number in the offspring. And to do that, you need to cut the amount coming from the father and the amount coming from the mother in half, so when you put them back together, you're back to a full whole again. Okay, whereas you don't worry about that mitosis. Where do you see mitosis? Well, virtually every cell in your body except for most nervous system cells, because they generally do not make copies or replicate. And the other place that you're generally going to see this is in well, just about every cell in your body except for the sex cells, right? I mean, that's really what it comes down to. And again, like I just mentioned, the nervous system cells. Now, meiosis, you're going to see predominantly in the sex cells, or only in the sex cells in almost all species. And again, because you have to cut the number of chromosomes in half, you don't want them to just be duplicated, because then each successive generation would pick up more and more and more chromosomes. So first generation would have like 46, and the next would have 92, and the next after that would just keep increasing, and so on and so forth. So you don't want that to happen. You want to keep it consistent throughout the species, so you cut the number in half. In fact, if individual were to have that many chromosomes, they probably would not be a viable candidate for birth. Okay, random assortment of chromosomes in meiosis. Here on the left side, you can see the organization of where each is located. There you get the purple and then the blue. And then in row 2, you have the blue and then the purple. And then row 3, you have the purple and the blue. If you look at the second photo, the one to the right, you have a change, and the first row, the light blue and the purple have alternated spots, so you get a different set of random assortment. And the third one, again, you get some additional random assortment, where the second row, the light blue and purple change positions. And so therefore you have a third option. Then you also have the fourth option, where they both totally align, independent of each other. Okay, here we talk about crossing over, how one sister chromatid, non-sister chromatid can cross over with another non-sister chromatid. So this causes quite a bit of diversity in individuals and who gets what. This is why you're not a exact copy of your mother or your father. You're kind of a combination of the two, because there is this physical exchange of chromosome segments, and crossing over generates new combinations of mom and dad's alleles. Okay, here we're looking at meiosis and what happens with the numbers for the haploid count, for instance. So here you have the primary spermatocyte, and you have the 2n value, and then you go to the secondary spermatocyte, which cuts it in half, so you go down from 2n to n, and then meiosis 2, you then start to duplicate those. So your spermatids, you now have a good bit of variation, and then finally at the last final step, each of those 4 sperm are genetically different, related, but different from each other. This is just an enlarged, or this is actually a photo of what's going on the female side of the house with the primary oocyte. The previous slide was taking a look at spermatocytes. This is looking at the oocytes. Again, the 2N is going to go through, break down into N at the secondary oocyte, and you also get these polar bodies, which will actually kind of disappear at one point in time. Then you have meiosis 2. You'll have some additional polar bodies made as well, still maintaining just that one egg. And finally, at the end, you just have one egg and the polar bodies kind of dissipate. Keep in mind that meiosis maintains a constant chromosome number from generation to generation. The formation of gametes. Meiosis results in two kinds of haploid sexual gametes. Males produce spermatids by the process of spermatogenesis, and females produce oocytes by the process of oogenesis. Spermatogonia and the testes divide by mitosis to produce spermatocytes. Spermatocytes undergo meiosis to form spermatids, the haploid value, and then spermatids undergo structural changes to become functional sperm. Okay, I think that was just the same picture again. Okay, oogenesis, this is for the egg. Oogonia divide by mitosis to form primary oocytes. The primary oocytes undergo meiosis to form secondary oocytes, which are haploid, and division of the cytoplasm is unequal. You get one larger functional gamete, which is the ovum, and then you get these three smaller polar bodies that do not have the equal amount of cytoplasm given to them, so they become these polar bodies. Products of oogenesis. Oogonia, mitotically active cells that produce primary oocytes in the ovary. The secondary oocyte, which is the large cell produced by the first meiotic division. The ovum, which is the haploid cell produced by meiosis that becomes the functional gamete. And polar bodies, the cells that contain little cytoplasm and will not function as gametes, produced in the first or second meiotic division in female meiosis. Again, there's that same picture of what's happening from the primary oocyte to the development of the egg. And then this is a great table comparing spermatogenesis and oogenesis. Again, this is something worth knowing. What's nice here, if you look at spermatogenesis, everything's just like 16 days apart. If you have the 16, 16, 16, 16, then you get the full 64 days to maturity. For oogenesis, again, 12 to 50 years is the total time for the egg to mature. Remember, a lot of these are formed in the embryo, so the eggs are around for most of a lifetime intact. And over time, depending on at what point in maturation of the female does that egg drop is totally random and unknown, but again, you'll get after 12 to 50 years that mature egg zygote total time, 12 being the earliest, and again 50 years being the longest. And that's the end of chapter 2. Thank you for paying attention. Hope that was helpful a little bit. And please, again, continue to read the textbook, and I think that'll help you, and hopefully this will give you a little bit of guidance on some important features. Thanks so much, and talk to you shortly. Bye-bye.